देन यू कैन राइट इट और ये प्रिंट आउट निकाल देता है। should I say?
Good afternoon to all respected dignitaries, our Director, Professor Vivek Lal, guest speaker and orator for today, Professor Subrata Sinha, ex-Dean and Head of Biochemistry Department, All India Institute of Medical Sciences, New Delhi, our Dean Academics, Professor Rakesh Sahgal, our Medical Superintendent, Professor Vipin Kaushal, all other dignitaries, Head of Departments, faculty members, colleagues, ladies and gentlemen. On behalf of the Department of Biochemistry, I extend a warm welcome to all of you to the 10th Professor R. Nath Memorial Oration. We will begin by welcoming our dignitaries with a floral bouquet. I'll request to kindly present a bouquet to our orator for today, Professor Sinha. I now call upon our director, Professor Vivek Lal, to kindly deliver the welcome address.
So, Professor Sinha, I apologize. The bouquet should first have come to you and then to me. So, please, sir. So, I welcome you all to this 10th Professor R. Nath Memorial Oration. Whenever I come here, they give me something to speak from. But I always fold this and I keep it here. I've never spoken because I guess I'm just one of the few lucky ones who's seen PGI grow, who has seen the gravitas and who has seen the substance and who has seen the divinity which PGI represents. Yes, that's the word, gravitas, substance and divinity. And you see, when PGI was inaugurated in 1962 or 1963, 7th July, Chandigarh was just a jungle. There were 2.5 sectors here, 2 or 2.5 sector honge. And this institute was built in a jungle. But by dint of the hard work, the commitment of forefathers like Professor R. Nath, this institute achieved a trajectory that has taken its alumni to the who's who list of the world. Professor R. Nath was amongst the first professors of biochemistry here. I think he was the professor and head, first biochemistry professor and head, if I'm not wrong. Oh, maybe. And after me, I think Professor Khulla followed. He did his fellowship in biochemistry from California. That's right, San Francisco, California. That was in 1956 and 56. And doing a PhD then, you know what it meant. You can compare that to today. Actually, there is no comparison. It's an odious comparison. But he came back. He joined as a faculty here, stayed back. And he was one of the founding fathers of this institute. I'm not saying the Department of Biochemistry. I'm saying one of the founding fathers of this institute who spearheaded the progress of this institute, which took us to another level, and that's a level which I have experienced partly. I must honestly confess, when I joined this institute, things are a little different now. He was, I don't think many of us, especially those sitting at the back benches, know what he was and what he looked like. He never smiled. I never saw him smiling. I was a resident. I joined this institute in 1987. It was frequent and routine for him to come and sit for CPCs here. He would always come, stoop posture, serious demeanor. You wish him, you'd get a response, but always without a smile. He was always thinking. He was exceptional in his vision. If you look at his achievements, besides what all he did, he actually started looking at trace metals before we realized their detrimental potential. He formed this integrated society for trace metals, looking at heavy metals, before we actually realized their detrimental potential. Today I can tell you 50% of toxicity of the nervous system is directly or indirectly related to trace metals and heavy metals. So he was far ahead of his times. His son Avinder was one of the previous orators who now heads the division of neurovirology in NIH. He is a director there at NIH. He gave one of the prior orations, and he actually was, when his father was ill, Professor Nath was ill, I was a resident who would go to meet him regularly, although he was not directly under me. He had a glioblastoma, and he was admitted under neurosurgeons. And I would meet his wife daily. His wife was such a pious lady. She was such a pious lady. I would go in the evening before I would go back. I would go and meet him. She would be waiting. She knew the outcome. There were no high-flying drugs at that time. We all know the prognosis of glioblastoma multiforme. But she never, ever showed any resignation on her face. We lost him. But that giant lives on through this oration. To give that oration, we have another giant today, Professor Sinha. He's a J.C. Bose Fellow. So thank you so much for coming, and is chair of the National Science Commission in Delhi, New Delhi AIIMS. Thank you so much, sir, for coming. That is very kind of you. And I think your presence here does, I would say, justice to the memory of that icon. He was an icon, one of the few face. When I saw this face, I didn't see this face actually. I saw him in a more aged avatar. He had aged by the time I saw him. And this is what he actually looked like, or he would have looked like when he was young. Always quiet, always helpful, never controversial, never controversial. I was talking to his students during lunchtime. I had already formed my opinion. This speech which I speak has been prepared before lunch, and they were telling me that how helpful he was. And that between 1 and 2 o'clock, he would lock his room. So that's something I never knew. And he would go and read the latest journals in the library. I do not know, many of you know there is something called a library here. I do not know. That's so sad commentary. When I went to inspect the library the other day, it was empty. It was empty. It was actually empty. It was very painful to see a library being empty. I know you would be studying, but the charm of studying in a library is something which only those who study in the library can actually, feel and, feel what it means studying in the library is. So with that, I welcome you, sir. It's an honor once again to have you. And it's an honor to celebrate his memory. He was an icon, one of the founding fathers of this institute. And I hope that this institute continues to prosper under his vision, with the vision of many more who may follow. Thank you.
Thank you, sir, for your excellent welcome address. I now invite Professor Sadhna Sharma, Head of Department of Biochemistry, to kindly introduce our orator, Professor Subrata Sinha.
Thank you, Dr. Deepi. So, respected Director, Sir, Professor Vivek Lal, Professor Rakesh Sehgal, Dean Academics, Dr. Ratho, Sub-Dean Research, Dr. Vipin Kaushal, MS of our institute, my faculty colleagues, residents, staff, and students. So it is my profound privilege to welcome Professor Subrata Sinha, who is one of the well-known personality worldly known for his outstanding achievements in medicine and research. He has got this significant contribution in the field of neurosciences and molecular oncology. He is the former Dean Academics, Dean Research, and Dean Examination at All India Institute of Medical Sciences, New Delhi. He did his MBBS and MD from AIIMS, New Delhi, and he availed several postdoctoral fellowships from India and abroad. He joined Department of Biochemistry, AIIMS, as faculty in 1988, and he achieved various academic and research positions after that. He has been Dean Academics, he has been Dean Research, and he has been Dean Examination as well. He has been the director of National Brain Research Center, which is one of the autonomous institutions of the Department of Biotechnology from 2010 to 2017. Presently, he is a J.C. Bose Fellow and the National Science Chair of SUB, which is one of the most prestigious affiliation being awarded to the person with an outstanding contribution towards medicine and science, which he is availing at AIIMS New Delhi. He is recipient of several awards and honors that include Swaran Jayanti Fellowships by DST, Fellow of the National Academy of Medical Sciences, Indian National Science Academy, and Indian Academy of Sciences. Being chairman of COVID Diagnostic Committee at AIIMS, he administered several research projects on COVID-19 that led to the publication of so many significant journals which are PubMed indexed. He is instrumental in developing a multidisciplinary collaborative research program on the genetics of glioma and is pioneer in the recombinant DNA technology, antibody technology. He has done significant work on the existence of multiple endophenotypes of dyslexia, and he has more than 200 publications in the peer-reviewed journals. So with this introduction, I will invite Dr. Subrata Sinha to deliver his oration.
Good afternoon, and Honorable Director, Sir, Honorable Dean, Honorable MS, Head of Department Biochemistry, Dr. Sadhna, Dr. Ratho, all other faculty members, students, and researchers, friends. It is indeed a great honor to be called upon to deliver the Professor R. Nath Memorial Oration. I was actually very recently inducted into the faculty of biochemistry when we used to, as a young assistant professor, he is an exalted figure. So you really, we used to look upon him with awe that, okay, he is the head of, senior person, head of PGI Chandigarh. I know about his work on trace metals. And more recently, when I was director of the National Brain Research Center, then we had, like you had his son for an oration, we also had an annual day oration for which Dr. Nath had come. And that family really supports research in terms of they have donated some awards for travel fellowships to Indian Academy of Neurosciences. So they, even though he is out of the country, he is very much, his son is very much involved in promoting our, research in India. So I would, can I have a pointer? Yeah. So the topic of my oration today is a very interesting aspect on dyslexia. Now, before I joined NBRC, I had no clue about dyslexia. I mean, I had a layman's clue about dyslexia, but not more than that. But there, so I work primarily on glioma, and most people have here who are in biochemistry and molecular biology have heard me talk about glioma. But over there was a program going on because a lot of this work has also been done at the National Brain Research Center, and this is now I'm continuing with it. So say, this is at Manesa. It's a DBT institute. So this is of course about Professor Nath, whom I have said. So basically, what is dyslexia? Dyslexia is a form of learning disability. And this work, first, before I start off, let me tell you who our collaborators are, because they had to teach me about dyslexia. So there's Dr. Nandini Chatterjee Singh. She's a physicist who's gone into cognitive neurosciences, who does neuroimaging. And she had this whole families of dyslexia, which has been documented with the help of Maharashtra Dyslexia Association. So this gives an opportunity to study. Mitali Mukherjee and Mohammad Farooq of IGIB, at that time when I started off, NGS was new, data analysis was new, so they have taught me NGS. Dr. Pankaj Seth, he has a wonderful model of human neural progenitor cells, which actually can differentiate from progenitors into neurons in the dish. And these are made from aborted fetuses. And this really provided a model for stem cell differentiation. And molecular modeling has been done by Dr. Deepti Jain of Regional Center for Biotechnology in certain areas. And essentially, I'll be talking about three families and drawing lessons from that. Three families with dyslexia, outwardly all looking the same, but the molecular mechanisms in these families being very, very different from each other. And each of these families has been sort of the work has been spearheaded by one PhD student, Tista, another PhD student, Bharat, and Shubha is our postdoctoral fellow who looked at the another family. So what is dyslexia? Dyslexia is a specific for neurodevelopmental disorder and refers to difficulty in reading and writing despite normal intelligence and adequate education. So please remember the catchword is normal intelligence and adequate education. So in spite of that, there is some specific circuitry problem because of which you cannot read or write as efficiently as another person can. And it has a worldwide prevalence of 5 to 10%. And there is a biological basis to it, but the exact nature of the pathways are not known. Essentially, what you are seeing is what is called dysregulated phonological processing, sound, letter, concordance, and meaning. This is deficient. And so they do, there are psychometric tests. You know, sometimes we think, as a biochemist, we tended to think that, okay, the psychometric tests, but they actually are not that reliable. They're actually pretty rigorous, pretty statistical, and pretty well derived. So, and like everything else, like all neurobehavioral disorders, it is multifactorial, has multiple etiologies, and finally manifest as a result of gene-environment interactions. So there are multiple susceptibility genes which have been mapped on GWAS and many other things, but worldwide the preeminent role of one set of genes is not very clear in dyslexia. It's likely to be a basket of disorders with a broadly common phenotype. So therefore, what is an endophenotype? An endophenotype, which is a phenotype which sort of resembles perhaps other similar disorders, but which has a particular specific genetic etiology. We can sort of extrapolate it to say that perhaps, you know, in glioblastoma can arise because of multiple molecular mechanisms, but outwardly to a histopathologist, a glioblastoma looks the same. So now in the current diagnosis, you also want to put in molecular classification because glioblastoma A caused by a subset of disease genes A will be different from a glioblastoma caused by B, which is a subset of disease genes B, and they too will be requiring different forms of perhaps specific therapy. So same way, we have not gone that far in dyslexia, but it is in many other psychiatric disorders in neurology, mental health, etc., where you have a common syndrome which have a specific, different, distinct genetic basis. So the other thing that I want to tell you is family studies. Now, India is a great place for doing family studies on extended 3-generational families because of our endogamy. Endogamy can be geographical, it can be cultural because of caste and tribes, religion, ethnicity, essentially the tendency of people to marry within their ethnic groups. And this is something which is still, even to this generation, quite prevalent in India, and earlier on it was even more. So as early as 1868, in the Indian Medical Service, there's K. MacLeod, who wrote in the Indian Medical Gazette that there are so many cases of individuals whose life and ancestry are confined to so small an area. And this can be obtained from people with stationary propensities, not possessing facilities or inclination to migrate. And he wrote at that time, the time when such records can be easily collected is rapidly passing away. Education and enlightenment combined with increasing facilities for migrating will eventually break down this barrier. But let's look at it a bit later, sorry. क्या हो गया? In Nature Genetics in 2017, from CCMB, there was Lalji Singh and Thangaraj, there was a Harvard group. They identified 81 unique groups, 14 of which had estimated census sizes of 1 million that descended from founder events more extreme than those of Ashkenazi Jews and Finns, which are the most inbred communities in in the West. And so they had that recessive diseases are also likely to occur at an elevated rate, even in non-consanguineous cases because of recent shared ancestry. So when we have a group of people who are marrying within their community, we can actually find traces of the family, even though they are not classically consanguineous. So I will be talking of 3 extended families, 2 in more detail and 1 little more superficially. So this first we managed with the help of wonderful volunteers from the Maharashtra Dyslexia Association to obtain the phenotyping and the results of an extended Maharashtrian family. And this is the pedigree. So you can see, of course, these we know from records. These we have, nobody has tested or encountered. But this is the first generation, second generation, and third generation. These are large extended families. Some not classically inbred, but within the same caste. And so we took a dyslexia cutoff, and there were families from which DNA was available, and families from which testing was available, and some number with both. So 76 people, 53 DNA samples, IQ and assessment 38. So we got a number of 14 cases and 24 controls. And this is the tests they have for dyslexia. In interest of time, I'll not really go into it in detail, but it's quite statistical. This is non-word reading. This is the random numbers. This is rhythmicity. This is phoneme replacement. So you can see that the controls and tests differ from each other. And this was a methodology of genetic studies. Some of them we selected for exome sequencing. And from then, we identified, because at that time when we started about 10 years ago, exome sequencing was expensive. So we identified certain SNPs and indels using different inheritance models. And then we validated with Sanger sequencing and amplicon resequencing in the rest of the people in which all the data was available. So we got 4 of them, 4 SNPs, which were statistically segregating with dyslexia. And the first one I would like your attention is on chromosome 5, brain acidic soluble protein 1, and there is an insertion in this from A becomes AGT, and this is located in the 5' untranslated region of a gene. So we decided to study it. There are other transmembrane channels, nucleosome assembly proteins, and another sensitive gene which we haven't really worked out because. And this is the ratio of so the maximum degree of kind of statistical significance you are getting after amplicon resequencing is with this BAST1 gene. So now something more interesting came up. This insertion is present upstream of the brain acidic soluble protein gene. And as you see, this is the protein gene. But one thing caught the eye of my student. I must say I had no credit in that. But the point is he was very good at bioinformatics. This is a lesson for everybody. Please, there is a depth of the world in bioinformatics, which we are not able to delve into, learn it, otherwise we'll all become dinosaurian. So this is a long non-coding RNA, which is transcribing, the coding gene is transcribing in this direction, the long non-coding gene is transcribing in this direction, and this has an overlapping region, which is 599 base pairs long. So the insertion is present at a 350 base pair downstream to the start site of this mRNA. So this is the insertion. And then this is what is now known as a divergent LNC RNA pairing. So there is a coding and a non-coding gene which are paired with each other, which are called divergent long coding RNAs. And what is the BAS1 function? BAS1 function was known that it is supposed to help in neurite outgrowth and synapse formation. And along with co-expression, you actually overexpress it in the artificially. It can elicit axon extension by adult dorsal root ganglion cells. These are mouse cells, these are not human. And overexpression again stimulated neurite growth into PC12, which are basically this adrenal medullary cells. The other thing that we know of is that there is a great neurodevelopmental role for long non-coding RNAs, and I'll not really go into that. But antisense long non-coding RNAs pairing with a protein coding gene on the opposite strand to regulate epigenetic transcription, silencing transcription, and mRNA stability. So then we decided, let's find out, let us characterize, because this is a gene which is so closely segregated with dyslexia in an autosomal recessive manner. That's what I forgot to tell you. This is associated in an autosomal recessive manner. That was the thing. So 2 copies has dyslexia, 1 copy or no copy has no dyslexia. That way. So we looked at the coding potential of this long non-coding RNA, the expression, and the effect of that on BAS1 expression. So we essentially, when you look at a long non-coding RNA, there are features in the sequence which tell you whether it is a coding sequence or a non-coding sequence, and there are various scores or average Pickett score and all that. This is experimental. Long non-coding RNAs are likely to be nuclear in localization. So this is the cytoplasmic and this is the nuclear distribution of the RNA in neural progenitor cells, which was we had obtained with the help of Dr. Pankaj Seth's lab. You see, gap DH that you all use is cytoplasmic, but the unspliced gap DH is nuclear, so this is a kind of a control. And then the BAS1 AU1, that LNC RNA, is also nuclear. And nuclear RNAs do have a kind of protein coding potential. And then, we saw the expression in, now there's Allen Brain's Atlas available, GTEx available. We find that it is expressed well in the brain frontal cortex, in the brain cortex, also comparatively well in the whole blood. In situ hybridization from the enhanced brain bank showed that it is expressed in the brain. And then in different kind of brain cells, both the BAS1 gene and the non-coding RNA gene are also expressed. This is the neural progenitor cells where both are high. In the neurons, some quite a bit less. Both this is the coding gene, this is the LNC RNA, coding gene, LNC RNA, and the astrocytes even still less. So now the question was, who regulates what? Do they regulate each other at all? And this is from the neural progenitor cell culture, which are primary cultures of human neural progenitor cells, which are obtained from fetuses, aborted fetuses. So you see, when you knock down BAS1, what happens? When you, this is scrambled, this is knocked down. So when you knock down BAS1, obviously BAS1 RNA, when you knock down BAS1, AU1, the long non-coding RNA, the level of the BAS1 RNA also falls. So when you knock down the non-coding RNA, protein coding RNA falls. However, when you knock down the protein coding gene, nothing happens to the long non-coding RNA. So long non-coding RNA is upstream and regulating. And then we looked upon its role in early neurogenesis. So we looked at the effects essentially by knockdown on proliferation of human neural precursor cells, on neurite length, etc. And to assess this, basically human neural progenitors were pushed towards differentiation. They were exposed to media supplemented with BDNF and PDGF to initiate neurogenesis, or with serum to initiate astrogliogenesis, and differentiation was carried on for 21 days. And RNA was extracted at different stages during the differentiation, day 1, day 3, day 7, day 21, and the LNC levels, etc., and the BAS1, of course, were analyzed. And what do we see? When you differentiate a progenitor cell towards neural or towards astrocyte, different things happen. When you initiate, so this is black is neurogenesis, this gray is astrogenesis, and this is the progenitor, which is constant. So on day 1 of neurogenesis, there is a marked rise of the long non-coding RNA as well as the coding RNA. And then this is followed by a fall. And this fall is quite marked, and it lasts. Both this is for LNC, this is for protein. In astrogenesis, it falls, and it remains low and remains low and it remains low. So in both the cases, so we were interested in neurogenesis. And then what we did was we knocked down the RNA in the progenitors on day 1, day 2, on day 1 and day 3, and we looked at the DCX plus cell count, which is basically the neuronal differentiation count, and we looked at the proliferation marker. And we looked at the proliferation marker, which is Ki67, and we looked at the DCX. And when you knock it down, knockdown reduces the number of neurally differentiating cells. This is the statistics, this is the picture on day 1 and on day 3. It increases the number of proliferating cells. So less differentiation on knockdown, more proliferation. And this is followed by a reduction of neurite length as well. So neuronal differentiation is actually hampered when you knock down this long non-coding RNA. And what we try to get some idea of the mechanistic way by which this happens. We haven't done it very rigorously, but one interesting thing I'd like to show you. This is called chromatin isolation by RNA pull down. How does the RNA actually regulate the RNA coding gene? So this is called CHR. So what you do, you cross-link the whole DNA, you sonicate, and you hybridize it with multiple biatylated tiling oligonucleotides, which are labeled with biotin. And these are the number of tiling oligonucleotides that we generated, 1, 2, 3, 4, spanning the whole length of the RNA, the long non-coding RNA. Now, I didn't tell you this, or I missed it when I was telling you. The RNA, long non-coding RNA gene coding on this side is relatively small. On the other side is the one gene, which has 2 exons, but separated out by 57 kilobases of introns. So basically, there is the 2 coding ends are far away from each other. And when you pull it down, with the BAS1 AU1, so you essentially have a streptavidin coated biotinylated bead, pull down whatever is bound to these dialing oligos, and then you do your real-time PCR, which will give you the extent of enrichment. So obviously, as expected, with all of them, the long non-coding RNA is coming down with all. But when you do a PCR of the BAS1 DNA coding region, in that case, R3, which is here, R11 and R14, which are here, so they come down. So the middle part is missing. So it seems that the RNA is binding to the two ends of the long non-coding of the gene. And this is a kind of simplified picture that the gene is this is the intron, this is the two coding region exons, the mRNA is here, and this somewhere is helping the gene to transcribe. So this kind of loop structure is important in the sense of DNA or transcription of the coding gene. And then, so this is one aspect. So RNA and DNA are forming a complex. Now, there is also again a lot of databases available. One of them is the ChIP-seq database. With the ChIP-seq database in neurons, suddenly we find that there is one interesting gene called TCF12, transcription factor 12, which is important for neuronal differentiation. And this is there in the database. So we try to repeat the experiment in our neurons, neuronal progenitors. And what do we see is that when you pull down with, do a normal chip assay, you pull down the RNA DNA protein complex with the antibody to TCF12. So pull down the protein. So all the DNA attached to that protein also comes down. And then you do a PCR. And then you find that another fragment is really enriched with TCF12. And this is, again, when you knock down. So what happens? TCF12 becomes interesting because previously in inherited dyslexia, one gene has been identified, which is called, this locus is called DIX1. So DIX1, C1 and TCF12 are coded by this locus. And so TCF12 is also binding to the complex. And when you actually knock down the BAS1 when you knock down TCF12, again BAS1 gene coding gene goes down. So what we've managed to do here is, with this family, identify one endogamous family with a GT insertion on 5p 15.1, which has the long non-coding RNA and RNA. So this is there from our study. In the other study, there is a DIX1 locus on chromosome 15, which is highly replicated in inherited dyslexia. And this causes TCF12. And we find that both of these come to one common pathway, which is critical to neuronal differentiation. So neuronal differentiation has a key role in dyslexia, especially in the endophenotype of this family. And two elements of inherited dyslexia can help in coding, in identifying this thing. So answer is, the same phenotype may result from the effects of different molecular pathways. So this phenotype is caused by low BAS1 and so on. So this is one sort of, in a way, our take-home lesson, apart from the fact that this is a unique pathway of neuronal differentiation, which this family helped us to study. So why do I say from behavior to pathway? You get behavior, you study a family. That family may not, that genetic defect helps you to find one pathway in that. Now that pathway may be widespread, may not be widespread. That is, may not be the case. But in this particular family, this is a biological gold mine in the sense that it will help us identify a new stem cell differentiation pathway. So many times you may have isolated families with isolated genetic predispositions, which may not be, it may not be that frequent for you to put it for a clinical test. But on the other hand, well, for that particular family, yes, it is important, especially for following up the family, which I'll come to next. Or else it will help you in identifying other things. So one thing is very clear that some forms of dyslexia do occur because of quantitative defects in stem cell differentiation. Now, why do I say quantitative? Well, obviously, if you have a drastic defect, you will have global neurodevelopment loss. So that is not a definition of dyslexia. In dyslexia, your global developmental parameters are more or less normal. It's a specific defect which is caused by this kind of pathway. And we looked at the probable molecular pathways which are common to all three. So what we did, we knocked down the LNC RNA, we knocked down the protein, and we knocked down the coding gene, coding mRNA, we knocked down the LNC RNA, we knocked down TCF12, and we find that common to all of them, there are three important genes, NeuroG2, NeuroD1, and HES1, which are very, very important for neuronal differentiation. So any of these defects of any of these can cause an aberration in that particular pathway. Okay. So take a deep breath. We're going very briefly, and I know that I must be brief, into another kind of pathway, but this is one I explained in slightly more detail because this is something which might be of interest to a molecular audience. This is another family. Between us, this is sometimes I'm always at a loss whether to mention the genetic group that you are getting these things from. Some people do. I know that people say that we got this in a Banya family, Brahmin family, in this family, that family, but I usually will not be writing it. I just write it as an endogenous group from West India. So anyway, so this is another endogenous group which is very, very highly involved, very, very highly involved. And here, what we find is dyslexia is inherited in an autosomal dominant manner. Totally different kind of inheritance as the other group. And here again, this was done by Pista, and these were the marked individuals which were chosen for exome sequencing, and then of course we verified it by our. And there again, reading, rapid naming, rhyming, phoneme replacement after ruling out intellectual disability and lack of opportunity. In fact, this is a very well accomplished family. One thing that people should realize is that it is worthwhile diagnosing dyslexia early because remedial measures can be put in, and there are some very, very creative dyslexics who have done very, very well. So sometimes, people say, badmash bachcha hai, interested nahi hai, ye hai, wo hai. Many of these may have a slightly biological cause which the parents need to be sensitive about and may need to really put in adequate, appropriate remedial measures early on. So this is an interesting family. They are very well off. I'm off record, it's a Parsi family, very, very well accomplished. So what we did was that we selected the samples, we sequenced, and we used two kinds of methods. And then we validated as usual. So we did whole exome sequencing and whole exome SNP array. And we got some results with this, we got some results with this, and we chose, because there are so many results which were coming up, to use the two results which were common to both. So firstly, the first thing that I was talking about, which was closely segregating in this autosomal dominant manner, is this. 17 variations of this in chromosome 5q 13.3 co-segregate with dyslexia following an autosomal dominant model. And this is the risk haplotype, this is non-risk haplotype. And even though this stretch of DNA is very long, for some strange biological reason which I have not been able to put my finger on, there is no kind of crossover in this one. So basically most of the variations are in this P2DH alpha, P2DH alpha, the protocadherin alpha. Now what is a protocadherin? A protocadherin there are different kinds of protocadherin. Studying protocadherin and its variations is a huge sort of thing. But they consist of a large variable region with a short constant region. And they are exposed on the surface of the cell. And protocadherin cell-cell interaction is important for neuronal migration. So neuronal migration is another neurodevelopmental pathway which is important, and which this study comes out with. So I will not really go into this in too much of detail. There were these 7 and they were distributed among different protocadherin, and these are the sites where they are distributed. I'm not really going into that, but essentially you have one risk haplotype which is variation, one non-risk haplotype which is variation, and these variations, some of them had non-synonymous protein alteration variations, 4 of them. Isoleucine to leucine, alanine to threonine, arginine to proline, and lysine to glucose. And so these did affect the processing of the protein as well as their kind of hydrophobicity and hydrophilicity. Plus you had in the RNA you had some variation. But why I am saying this is not so much to talk about the variations, because it's very difficult to study this complex protein in detail in the lab. But you see this. So there are variations which are in, so we have dyslexic and we have non-dyslexic in this one. And there is this variation which is predominant in the normal readers, and there is this variation which is predominant in the dyslexic. And there are certain variations which we see which arise only in the Homo sapiens, and now they are not there in the Homo neanderthalis, which is the Neanderthal, or the Denisovans. And there are 7, and they go all the way down, some of them to chimpanzee, to gorilla, to macaca, and so on, and mice and rabbits as well. So there are, so this is lineage specific variations are present in PCDH A3 locus, A4 locus, A5 locus, and C3 locus. And in all of them you see this one, and then you see this. You see here, you see it goes on to here, and then you see this. And so in many of them, many of these variations, most of these are specific to modern human species. So, I mean, it's easy to speculate because this is, again, you see G and the rest is C. G, rest is C. A, rest is G. So, these are obviously, again, we are in the realm of speculation, a kind of evolution has occurred because of variation, because of this neuronal migration becoming more efficient, and this has given rise to cognitive functions which are associated with normal human beings. Because one thing I forgot to tell you, you see, reading and writing is a learned behavior. It relies on human plasticity, and all animals vocalize. So far, no animal has been known which actually converts symbols into a sound. So reading and writing is a human-specific behavior. And this, I mean, we can go back into archaeology, but apparently it's not more than 10, 15, 20,000 years old. So, of course, drawings have been there. So there you go really into speculation. But it is definitely something which, as of today's knowledge, is only there in the modern human. So these are certain aspects of genes which have evolved, may have. These variations are statistically very significant. And they associated with ADHD very time. So we took a family which had both dyslexia and ADHD, and we looked at either dyslexia or ADHD or both as one group, and the non-dyslexics as one group. And we do find, same process, we do find that exome sequencing identifies a 1.78 kb non-haplotype in a gene called PAM3A co-segregating with dyslexia and or ADHD. So I'll just rush through the results, but essentially it is chaperone, and here high levels of this PAM43A is overexpressed in the blood of subjects with risk phenotype. One thing, let me tell you, your human PBMC, your leukocytes have a lot of these receptors. Lot of the receptors for all your neurotransmitters. So leukocytes very often become actually a valid model for studying many of these interactions. So this is with this haplotype, you see this is the level, with this haplotype, this is the level. These are all the human blood from the affected and unaffected. This again is recessive. And then we did some experiments and find that when we do a luciferase assay, if we clone the variants into the luciferase, the expression of the protein is higher. And if PAM43 is higher, they are present, of course, heavy nuclear. This is anti-correlated to genes involved in GABAergic signaling. So in this is a database. I haven't doing database as well, but whenever different subjects, but whenever the expression is high, the GABAergic genes are low. And we did some experiments in SS5Y cells, which are neural cells, and we find that when we basically do a knockdown, the receptors go up, and when we overexpress, the receptors go down. So, and verify with qPCR data. So essentially, again, GAT2 is increased. So basically, the GABAergic pathway is adversely affected by the overexpression of this gene. So in this family, what we find is a neurotransmitter aberration, which is in head. Essentially that. So there are three different families of dyslexia. Each family has given us insight into one very critical aberrant pathway that was not known before. And this is why we must study. This is a way of getting to, in a way, a depth of the problem. I don't know how much translational value it has. I'm sure it will have something. All your North genes were Drosophila esoteric genes about 50 years ago when they were first discovered in Drosophila. Today, North is there all over. So wind pathway is there all over. Wind is wingless of Drosophila. So somebody studying flying for this. So basically what ultimately this molecular heterogeneity of apparently similar phenotypes will show us that dyslexia is shown up as a basket of distinct molecular changes. So it helps us in reclassifying and retyping a disease. And then eventually, I hope we will have to be, maybe we will be able to diagnose it earlier. The family which had that high, the autosomal dominant dyslexia, they tell us, well, we have a newborn child, can you please give us this genotype? Because we want to test and we want to put this child into remediation even before he or she learns to talk, so that we can do. Now, can we not? Is it ethical, not ethical? I really don't know. Finally, to be fair, we figured out we didn't keep it. We said, yes, it's not really a part to be used for clinical work. But please put him to remediation, because remediation will help even if he doesn't have, or she doesn't have dyslexia. So it will help you within phenotypes, within endogamous groups. If a mutation is prevalent, it helps you in diagnosis. It helps you in getting to the basic biology of the pathway. So this is something that we have a better understanding, and this is why we should study endogamous groups also for family studies. personalized book-specific management, feasible research questions, and we still have that resource very prevalent. And so, and another take-home lesson from this is please work together. All these people who do genetics, physicists, stem cell biologists, all of them have taught me, psychologists. I knew nothing of it before I started. Thank you very much.
Thank you, sir, for your brilliant, interesting, and highly educative oration, sir. Thank you so much. I would now like to read out a message which has been sent by the family of Professor R. Nath. You can project it, please. This message has been written by Dr. Avindranath. He is the clinical director of the National Institute of Neurological Disorders and Stroke at NIH, USA. So the message reads, the family is grateful to PGIMER and the Department of Biochemistry for hosting the memorial oration and in the name of our father, Professor R. Nath, who devoted most of his academic career to this institution. For him, the department was an extension of our family, and we have fond memories of the close interactions with the faculty and students, which also help shape our own lives and careers. We would also like to thank Professor Subrata Sinha for delivering the oration. Sorry we are unable to attend the event in person, but we convey our best wishes to all. Avi, Pamela, and Monica. I now invite our director and our dean to kindly present the memento and certificate to our orator, Professor Subrata Sinha. Thank you, sir. I now invite our dean, Professor Rakesh Sehgal, to kindly propose the board of thanks.
Good afternoon, everybody. And when jokingly, when we were having lunch, I was discussing that and telling him that maybe we can finish early because I have to go somewhere. But anyway, I think the way he has talked, I was telling him now that we could have gone even for an hour more and still we would have enjoyed. He has presented the thing so beautifully and it was a wonderful, talk and it was really wonderful to listen to him. So we are always thankful, sir, that you have come here and this you have enlightened us and well, because it's a lab related thing, so it's sort of near by what, though we don't do human samples, we do say parasites, but I really thank you very much. And I think whosoever has come here would have gained a lot of knowledge regarding what he has told. And I thank Director Sir who is here to grace his opinion. I think he would second my opinion that this was indeed a wonderful talk. And there is Professor Kaushal, then Dr. Ratho, and obviously the Department of Biochemistry, the head and other faculty who are here, and I see Dr. Reena also and other faculty. Dr. Sabhi बैठे हैं cytology के head. So, and I hope you had enjoyed it. At least I enjoyed it. So anyway, this is all I have to say, and I hope that we can invite him once again for some and in another occasion so that we have such an interesting talk. Thank you very much.
Thank you to everyone for attending the oration. I now invite all of you all to join us for high tea. Thank you.